Okay, I'll get started with just a little bit of housekeeping while a few more people trickle in. Welcome everybody to the 5th webinar in our KBA series. My name is Jamie Grimm and I'll be facilitating the meeting today. This webinar is being recorded and will be uploaded to our website tomorrow. You can also find all the recordings of our past webinars on our website. I will share the link for that in the chat box in a moment. So we have 3 speakers today who are going to be presenting on various data products and tools that we've been developing and using to identify KBAs across Canada. Many of these tools are open access and are going to be broadly relevant to other conservation products and projects rather in Canada. We'll ask that you please use the chat box function to ask any questions, which we'll save until the end of our 3 presentations today. At that time, when we have a question period at the very end, I'll also allow participants to unmute themselves if you would prefer to ask your question verbally, but we would prefer you use the chat box if possible for you. So, as I mentioned, we're going to have 3 speakers today, so I'll take a moment to welcome them. Chloé, Christine, and Juan will be joining us. Chloé is the lead data analyst for the KBA Secretariat hosted at WCS Canada. Christine is the national coordinator of the EBAR mapping project at NatureServe Canada. And Juan is a postdoctoral fellow at McGill University working with Andrew Gonzalez, and will be talking about a tool they've developed to identify the places that are most likely to be KBAs across the landscape. So with that, I will invite Chloé to please share your presentation and kick us off.
Thank you, Jamie. Let me know if you can see this. Can you see my slides?
Yes, perfect, thank you.
Perfect. Thank you, Jamie, and thank you everyone for joining. So today I'll be talking a little bit about the data infrastructure that underpins the Biodiversity Area initiative in Canada. And also I'll be presenting a quick example of how this infrastructure is being used by the KBA Secretariat for a KBA scoping exercise. And I'll explain what I mean by that. Okay, but before we start, I just wanted to provide a quick refresher on what KBAs are. So you'll remember that KBAs are sites that contribute significantly to the persistence of biodiversity. The specific criteria and thresholds for identifying KBAs are laid out in the Global Key Biodiversity Area Standard that came out in 2016. In the standard, specifically, they lay out 5 criteria that a site must meet at least one of these 5 criteria in order to be a key biodiversity area. The first criterion A, captures globally threatened species and ecosystems, such as species that are red listed. The second criterion B, is for geographically restricted species and ecosystems, for example, endemics. The third criterion captures sites with high ecological integrity, so you can think of those as having a low human footprint and having changed very little over time. Criterion D is for species that aggregate in large numbers, for example, during migration at stopover sites. And finally, criterion E captures sites with high irreplaceability through quantitative analysis, and the methods for this last criterion is still being developed. Accompanying the KBA standard, there's also a set of guidelines that outline how KBA sites can be delineated. A few reminders of what some of the key components of KBAs are, some things to remember. They can capture sites for any taxa and ecosystems. They can hold one or multiple qualifying biodiversity elements. They can be sites of any size within reason. They need to be essentially manageable. And all forms of knowledge are appropriate for KBA identification. What they are not, KBAs do not imply a management prescription, and they do not imply restriction in access. So really, KBAs are an informational layer that can help inform conservation decisions. They're a quantitative and scientific tool that tells us something about the importance of these sites. So as I've just outlined, the global KBA standard tells us how to identify sites that are of global importance to the persistence of biodiversity. And here in Canada, we're also developing a national KBA standard to identify sites of national importance to the persistence of biodiversity. So sites that help biodiversity persist nationally. And so this is the idea behind this national standard is to make the tool even more relevant to the Canadian context. And I won't go into much detail here, but I just wanted to present quickly a diagram of the organizational structure of the Canada KBA Coalition, just to give you a sense of how collaborative this work is and the number of partners that are involved in the process. And this is important for what I'm going to present next, because our entire data infrastructure is built around this idea of collaboration and how to enhance collaboration. So why a collaborative data infrastructure? Well, the Canada KBA Coalition produces a variety of data products ranging from the site delineations of the KBAs, including also the information that accompanies KBA proposals. We also track KBA trigger species and ecosystems. And Christine and Juan will also present some other products that they're creating, namely ecosystem-based automated range maps, or EBAR maps, and habitat suitability models. And of course, more products are being added to this list as the coalition grows. At the moment, these data products are being primarily created in collaboration among 3 partners: Birds Canada, NatureServe Canada, and WCS Canada. And finally, of course, we aim to share these data products as widely as possible, both with the members of the coalition at large, with our regional KBA coordinators, who are the people who are responsible for overseeing KBA identification in different Canadian jurisdictions. We want to share these products, of course, with NGOs and academic partners, with subject matter experts who help us review KBA proposals during the KBA identification process. We also share information with sites, stakeholders, rights holders, and other interested parties. And finally, of course, end users of KBA information also need access to this data. So this is the context for why we need a collaborative infrastructure. It provides a bit of a backdrop. What I'll present next is more of the actual technical infrastructure itself. I just want to say that this is still work in progress, and so some of these components might change in the future. But at the moment, a lot of the work of the KBA identification happens within what we call the internal KBA workspace and database. It is co-managed between WCS Canada and NatureServe Canada. It's a Postgres PostGIS database that is served via an ArcGIS server, and we access it through ArcGIS Pro, R, and custom applications. Within this database, there is really a wealth of biodiversity data that comes from a variety of sources. I won't go into too much detail here because I know Christine will explain a little bit more later about how the data was gathered, exactly what it includes. But for now, I just want to mention that there's a lot of data from different Canadian CDCs, conservation data centers, other government agencies, academic sources, museums, etc. And so different members of the coalition, different users have different levels of access to this data depending on their use cases, and also depending on the level of training they've done for handling sensitive data. What I'll call our internal ring of users have direct access to this data. These are the regional KBA coordinators. As I mentioned earlier, they shepherd the work of KBA identification in different regions. The Canada KBA Secretariat here at WCS Canada, as well as various academic partners. These different users produce various data products, including the identification of KBAs themselves. At the Secretariat, there's also a process to review the quality of the proposals being made. We conduct scoping exercises, and I'll present an example of that in a bit. And of course, you'll hear from Juan later about some modeling work that we also do with this data. These data products that are being created by users with direct access are then shared further with different users. So one example is we have a technical review app that enables species, ecosystem, and site experts to access some of the data products that we create to help us review KBA proposals and give us additional information about the sites and ecosystems and the species that are present. We also have a stakeholder review app that is customized for stakeholders, rights holders, and interested parties to provide information about KBA sites that are under review. And finally, we have a public progress dashboard that I would invite you to go visit when you have a chance, and that displays where we are currently looking for KBA sites throughout Canada in real time. So that is constantly being updated as we make progress. And of course, the other half of the database is where ecosystem-based automated range mapping happens. And I won't say too much about this right now because I know Christine will explain this in detail. But I'll just say for now that these EBAR maps then feed back into our own data flows and help us with KBA identification, and so also with KBA scoping, which I'll present in a bit. But of course, it doesn't end there. We want to share the data even more widely. And so the data that is, and the data products that are in this database then get shared in their final form with Birds Canada and their central KBA registry. The data that flows between these two databases is for non-bird species. Bird analyses happen in the central registry itself, and also for all ecosystems. Birds Canada will then be serving this data out to end users, in particular government agencies, industry, NGOs, the general public, etc., to make it as widely available as possible. And the final step that we hope to implement is to have both databases connect to the NatureServe Explorer interface to also serve the data to end users and provide another platform for access. All these data flows are being coordinated by the WCS Canada and by the Canada KBA coalition at large. So this gives you a sense, a little bit of how we've structured things and how data flows between us and what we're trying to do to make data as widely available as possible. Now I'll go into a little bit of an example of how we're leveraging this infrastructure to conduct what we call scoping for KBAs. So by scoping, essentially, you can think of it as screening pre-existing conservation sites to see if they have KBA potential. As you'll know from previous webinars, the process of KBA identification itself is quite labor and time consuming. And so it helps us if we can have some automated way to screen sites nationwide to see which ones may have high potential and which ones we might prioritize for the next step, which would be the more thorough KBA identification process. In this exercise that I'm presenting today, we looked at pre-existing important bird areas. So there are 582 Canada-wide, and we were interested in seeing whether they had any KBA potential for non-bird species, so for other taxa, as well as infraspecies, so subspecies, varieties, etc. We were assessing specifically whether there was any potential for these other species to meet KBA criterion A1, which is for threatened species, and criterion B2, which is individually restricted, geographically restricted species. Sorry. And we did that for both global and national KBAs. A quick sense of our method. So the idea with scoping is to be able to use widely available data sources to be able to capture as many species as possible. This differs with the KBA identification process that is more time-consuming and that involves identifying the best data source for each site. In this case, we have to essentially capture as many species as possible, so we use a broader umbrella approach. The data we use mostly is range maps because they enable us to estimate rather the percentage of each species global or national population that is found within the candidate sites. And particularly we use IUCN range maps, Environment Climate Change Canada range maps, and the EBAR maps that Christine will present. And because because range maps aren't sufficient information often to actually confirm that the species is actually present, we also use element occurrence data provided by conservation data centers to confirm species presence. And so with these data sets, we had 424 species and infraspecies that had sufficient data for this scoping exercise. Some quick results. So this is a national scale map. So I'll show you zoomed in areas in a little bit because it's hard to see at the national scale. But just to give you a sense here, we're looking at the number of elements, so species and infraspecies that met KBA criteria in pre-existing IBA sites using these methods. So the different shapes are existing IBAs, and the different colors show the number of elements from 0 to 14. And you see that some of the northern IBAs here have fewer species, and you can already tell a little bit from the national map that there are bigger hotspots in the southern parts of the country. We can zoom in, for example, in southern Ontario and see that there's, depending on the IBAs, much variation. Some sites are not capturing many potential non-bird triggers, and others, like Long Point, have several. Another example is southern BC, where you also see several sites that pop up as having potential KBA potential for non-birds. The next steps with these kinds of analyses is to then first expand the analysis to other pre-existing sites of conservation relevance in Canada, in particular protected areas and biosphere reserves. So we'll be looking at over 9,000 sites to be screened throughout Canada in total. We'll be looking to compare the results from this scoping analysis with the results of our more thorough bottom-up comprehensive approach that we take when you actually look to identify the KBAs to get a sense of how reliable these results are. And finally, we'll prioritize the areas with the highest potential for more thorough KBA assessment. So this is just to give you a taste of how we can use this infrastructure, and Christine and Juan will present other use cases. And just to wrap up, I just want to invite you to visit our website to get any information about aspects of the coalition that I didn't mention today, including governance, etc. Thank you.
Great. Thank you very much, Chloe. And just a reminder, if you have any questions for Chloe, you can put those in the chat box, and we will save all the questions to the question period at the end for all three of our presenters today. So Christine, I'll invite you to take over and share your presentation.
Can you see my presentation and hear me?
Yes, we can.
All right, great, thank you. So I'll be talking about the ecosystem-based automated range map project, also known as EBAR. Okay. Perfect. So as Chloe was mentioning, I'll just start off with explaining how the KBA project and the EBAR project kind of support each other. So EBAR is providing species level information that is being used to quantify KBA criteria. So specifically, as Chloe was mentioning, A1 and B1 require information on the proportion of a population at a site. And proportion can be inferred by range. However, range data is not available for many species in Canada, so we're really able to fill that gap, and not only for the KBA project, but also the greater conservation community in Canada. So how did we actually go about doing this? So firstly, I just want to acknowledge that our process is based on the work of the Northwest Territories, British Columbia, and Oregon NatureServe programs. But for our particular project, what we started with was data mining. So we went out and collected data from various sources with the goal of creating accurate range maps from the best available data. And I'll talk a little more about that later on in the presentation. So we then took all of that information and created what we call auto-generated ranges using custom-built Python code to populate ecoshapes with the species presence information. But because this is an automated process, we wanted to ensure that the resulting range maps were accurate. So we were able to do this by enlisting expert review. So these experts were efficiently and securely captured in the form of feedback using an online app that was based on existing code from NatureServe's map of biodiversity importance. So once we were confident that our range maps were accurate, given the current knowledge of that species, we are publishing all of these ranges. So they're going to be available online at no cost, both in PDF and geospatial data packages, and I'll provide you with links to how you can access these later on in the presentation. But a key component of the EBAR process is actually continual improvement. So we're hoping that through time, as new data or expertise is available, we can actually make improvements to the range maps using our existing infrastructure and apps. So what makes EBAR unique or different than a traditional range map? So if we kind of start with thinking about a traditional range, these are often point-in-time representations. They're created for a report or a publication. They're not intended to be living products. They can also be quite time-consuming to create due to the fact that the line work has to be refined based on the best available information. So you can imagine a lot of potential consultation about where a line for a particular species should lie. Should it be on this side of a mountain range or that side of a riverbed? That type of consultation requires a lot of time. And finally, traditional range maps can often have low transparency to inputs. We don't have a lot of information about what actually went into generating that range. So if we contrast that with EBAR, as I mentioned earlier, we have a lot of ability to have continual improvement anytime we have new data or expertise that's available. We've also automated a lot of the process using the custom Python tools that allows us to produce not only a range, but a huge amount of metadata that goes along with that range. And finally, all of our decisions are actually stored in our database. So if someone were to come to me and say, why is this ecoshape lit up but not that one, we actually have all that information available to have transparency. So how do we actually go about creating all of these different ranges? So obviously we need to start with data mining. So for those of you that are not familiar with us, NatureServe Canada is considered a national leader in biodiversity data management. So we were really able to leverage our existing partnerships with both governments and non-governmental organizations to engage with the wide variety of sectors that I've highlighted on the right of the slide. We were also able to add in additional data from online platforms using really easy to use R script. So we were able to download information from places like GBIF, Bison, and VertNet. We were then able to import all of that information from all of these different sources, again, using Python tools that allows us to easily and efficiently clean and standardize the data. So we were able to consolidate generally complicated things like taxonomy and dates and bring them all together into the internal KBA workspace and database. And as Chloe mentioned, this database was designed to be securely accessed from multiple locations because our team is really spread across Canada and accessed from a variety of platforms such as ArcMap, R, and QGIS. And I'm really excited to share that at this point, we have about 4.8 million species occurrences that we're able to pull from to do our work in the database. So how do we actually go about going from that data to the resulting range map? So I'm going to go through that in a little bit more detail. So the first thing we start with is our ecoshape mosaic. So we've developed that for Canada, US, and Mexico, because that's our general sort of geographic scope for this project. So this ecoshape mosaic is basically polygons that are ecosections, ecodistricts, or at their core, representations of ecosystems. So this really allows us to speed up our range map development process because we're starting with predefined ecoshapes that eliminate the need to draw or edit range boundaries. So now that we sort of have our base map, we add in the species data that I was talking about earlier. So that comes from that large scale mining exercise that currently includes the 4.8 million species observations. So all that information is buffered using the uncertainty distance from the data provider, because not all information is 100% spatially accurate. We then end up with our auto-generated range, which again uses Python tools to populate ecoshapes with one of three presence values. So the options there are present, presence expected, or historical. And then they go through the final process of being expert reviewed to see if there was potentially any issues in the data, either incorrectly digitized locations, or possibly in our data mining, we missed some locations. So again, all of that information is stored in our database, and experts are actually able to provide 2 levels of feedback. They can either go ecoshape by ecoshape and tell us that an ecoshape should be added or removed, or possibly the presence value should be changed. Maybe it was historical, but it was recently observed, so it should be changed to present. And then they can also give us the overall range. Based on the information that we have available, was it pretty good and we just have a little bit of edits, or was it absolutely horrible and we really should just go back to the drawing board for that species? So what have we achieved so far in our project? So as I mentioned earlier, we have about 4.8 million species observations that are coming from about 163 data providers. We have 1,264 auto-generated ranges. And before we send these out for expert review, the team always does a quick once-over just to make sure that there's no data gaps or errors. And I'm really pleased to share that we had 100 individuals contribute their time and expertise to expert reviewing our ranges. So I really want to acknowledge all of these people and just give them a huge thank you. And to date, we've published 163 ranges. We are continually adding to this number. I have about 140 that are waiting in the queue. So hopefully in the next few weeks, you'll be able to see them there. And you can view these on our website, our app, or NatureServe Explorer. I'll give links to those shortly. And all of the code that I've discussed in this project is actually available on GitHub. So we are always looking forward to potential collaborations. But if people want to see all of that sort of fine scale information, it is absolutely available for people to utilize. And I want to acknowledge the amazing team at NatureServe Canada that has contributed to the EBAR project. We wouldn't be here without them. And finally, this is how you can contact us. We have an email, we have a website, and we also have an online app where people can go to view and interact with the ranges. So thank you so much, everyone.
Wonderful. Thank you so much, Christine. And Juan, if you would like to take over and share your presentation.
Yeah, sure. Okay. Let me see. Second. Can you see my screen right now? Hello. I think it's frozen.
I
think you're good, Juan.
Can you hear me and see my screen?
Yes.
Okay, because mine is frozen. But anyways, thanks, Jamie, for organizing this and the opportunity to show our work and to help in this process of identifying key biodiversity areas in Canada. In particular, I would like to show the work that we are doing here at McGill University with Andy, trying to create tools to scope the boundaries of potential areas for KBAs in Canada. And I'm going to focus on 4 points now. The first one is going to be a little bit of background, how we started with this process, then the approach, a couple of slides to show the general approach that we are taking on this, and a demo. I'm going to stick with this PowerPoint because we don't have time to go live, and finally with next steps that we envision from this side. So I think a year ago we started with this idea of a big list of species that might trigger some criteria within these KBA standards and guidelines, especially A1 and B1, threatened species and individual species, and species with small geographic ranges. And I have to say that this list, around 400 species, is based on the consultation process with experts that they can see that that they can find information to fulfill the criteria A1 and B1. And also it's a list of the species of that can trigger like global triggers and also national triggers. Maybe some species are not threatened at the global scale, but maybe those species, some species are threatened here in Canada. So we thought that important to apply this process for these two groups of species. So the general questions, the general question at that point was if we can develop some methods and tools to help in this process of scoping the boundaries of candidate areas or sites for KBAs in Canada. So we envision like a top-down approach, more microecological analysis, because we have this list of 400 species, and also the extent of the analysis is North America, it's Canada and US, because some of the species go across the border, so we need to gather some data from US and Canada. And also we are going to work in parallel with the development of these data sets, the work that Chloé mentioned before, and also Christine about the EBARs. So, and we need to connect with these data sets to update if we have new data, and also replicate with a new species if we add new species to that list. So the scripts are basically in R, and we have the potential to connect with the data sets with NatureServe. But we also envision also a friendly and informative visualization tool in order to show the whole process, and I'll explain a little bit of that later. And we also want to include different needs from different users, from local, regional, national users, and researchers, and different taxonomic experts and decision makers. So I'm going to show you the tool in a little bit. But I'm going to start with the approach, and basically we have two big steps that I'm going to summarize here. And the first one is taking advantage of this habitat suitability model, because we have observations from the dataset that Chloé mentioned before within those EBARs, and we can use also predictors in order to predict habitat suitability for a particular species within those EBARs using an algorithm. So this one is a more detailed explanation of this. So we use these geographic ranges from the species, the EBARs, and the observations from the dataset that Chloé mentioned before. And we use a set of variables as a predictors, including topographic or heterogeneity, and some metrics of productivity, and some metrics of climate. And then we use for habitat suitability model maxing as an algorithm, because it has shown that some good performance with a small number of observations, but also because we have this package in eval package that allows us to evaluate different candidate models. And evaluate different components that are important for the final outputs. So as I don't want to go into detail, but for these data sets, we went through a process of cleaning the data. We selected we deal with collinearity between variables, and we have some modules here for model evaluation in order to have some kind of metric for potential usefulness of these habitat suitability models. The second step is based on these habitat suitability maps that we have here. We focus on these upper quantile and try to see if those high suitable areas are clustered in space. So we decided to run this density analysis in order to identify those hotspots, and that we have represented here in polygons. And we can see that some of these polygons here, and for instance, this one, this purple here, it holds more than 50%, 60% of the high suitability areas in within the geographic range of this species. So we have here this one, and the red one maybe it holds like almost 10%, and the yellow one like almost 20% of the high stability areas here. So those are the potential sites that can harbor in the future KBAs. Let me give you an example of the tool that we are creating. This one is the main landing page, and we run the analysis for 25 species or so, and this is an example of the final visualization tool for all the sites. This is just a draft because we are in the process of evaluating this tool, but let me just show you the four tabs that we have here. The first one is the workflow. It's explaining. We try to explain everything that we did in this process, and the first one is the workflow, that trying to go in detail about the process. And let me jump to the methods, because the methods is just basically explaining in detail what we did in these habitat suitability models, site identification, and additional modules that we are working on right now. I'm not pretending that you read all these, because these are just screenshots of the website. So, but if you click here, for instance, in data, so you can see what kind of what type of data we are using here, what are the process of cleaning the data, and if you click here, for instance, in model fitting, you can see how we deal with multicollinearity among those predictors. And in model fitting, it's very important that people understand these kind of how was the process of dealing with, for instance, the effect of background points of sampling bias. So people just can go back and read and have some links important to if they want to go in detail about these topics. But the most important part is the species report, because you can just search by the English name of a scientific name, and you can just select the species that you are interested in, and just click on and just is going to open a new tab, and we are going to have different tabs with information for every single species. But before going through these details, I want just to focus on this part here in green, that is telling about that this model in particular for this species seems to be potentially useful based on the analysis. But it's not the case for all of the species, because maybe we were able to run the analysis for the species, but maybe we want to highlight that there is some concern in model predictions of this model. And for other species, we don't have enough information to run the analysis, so the analysis just stopped there at that point. So we have different results depending of the data that we have for the species, but we want to show the whole process here. So let me go through these tabs quickly here. The first one is this interactive tool, where you have, for instance, in white the EBARs for the species, and in colors you have the polygons for those potential sites that hold most of the high stability areas within this range, and you have here the habitat suitability map for those species. But we have a bunch of layers that like users can use in order to make further decisions of this. Then we have this tab here, species status, where you can see that this one is a global trigger, and this one is triggering B1, which is a species with small geographic ranges and a lot of information that I think is useful for the user. Then we have the observation. We want to track the number of points, polygons, different features that are part of the observations, and here the process of cleaning the data and using finally, in this case, for instance, 29 points effectively used within the models. Then I'm going to jump from these predictors because it's just showing the predictors itself for that particular species. And then we have this habitat suitability model where you can just go through specifics of the model settings for that particular species in bold here, highlighting what kind of models and how we built the models for this particular species. And then we have also within that habitat suitability model tab, how we select the best model based on AUC, and here you can see that we test for different things that can affect model performance. At the end, we have the habitat suitability map and some stats, and we add also some uncertainty maps for these models. And the site identification that I showed you before, showing those cluster sites, but the top 3 that can hold most of the high habitat suitability with suitable areas within the EBAR. So this is the general tool that you saw before. Is a landing page, and then species by species that people can go through and just use this tool in order to identify and complement this tool with different approaches. This is my last slide. We're in the process of the evaluation, so we asked different people, including Chloé and Christine, to look at the usefulness of this approach, to scope the boundaries of potential sites, and also the validity of these data sets that we are using here, because we need to add some rules, so we need to be in the same page if we are using in the proper way these data sets. Also, we ask some experts if they see some important information from this macro approach. And we have to work with NatureServe and also WCS in terms of obscuring some sensitive data at the end of the process. And we ask some people also to look at in depth about the models itself. So we are going to make some adjustments here, incorporating the comments and suggestions, and we are going to run the analysis for 400 species to have this tool available for experts in order to complement with different approaches on this process of identify key biodiversity areas in Canada. That's all I have. Thank you so much. Welcome. Be happy to answer questions.
Wonderful. Thank you so much, Juan, and to Chloé and Christine as well for your wonderful presentations. If you have questions for any of our presenters or all of our presenters, please put those in the chat box, or feel free to raise your hand, the raise your hand icon, and I can unmute you to ask your question verbally. So we do have a few questions already, some which have been answered already. There's a great conversation going on in the chat box, but I'll confirm a few things verbally. Kate asked, how does one become a part of the KBA coalition or a partner? And Kira addressed that in the chat box. It is an open partnership. If you want to be involved, then feel free to send Kira or I an email. You can also contact us through the KBA website, and we can go from there. Dave Fraser has a comment that maybe one of you would like to address. He writes, EBAR range maps would be more useful if we could turn on the underlying records and EOs and have them show up on the map.
Yeah, so I guess I'll jump in there and just state that, yeah, we certainly envision EBAR as being part of a suite of products. So, you know, if you kind of think about it in terms of scale, you would have EBAR at the top. Then hopefully in the future, we hope to have HSMs like the product that Juan was talking about, then EOs and source features, and then all the way down to sort of like the precise species observations. And we would certainly encourage users to choose which one of those spatial scales is most appropriate for whatever application they are thinking of. And I also just want to say that we hope that in the future on NatureServe Explorer, you will have that ability to kind of see that full suite of products on that platform and application. So that's something that we're working towards. We're just not quite there yet.
Great. Another question regarding EBAR maps. Will EBAR maps update or replace the range maps available on the IUCN Red List?
So yeah, we haven't had any kind of discussion with the IUCN. So I would say there's currently no plan to have EBAR be integrated into the IUCN platforms.
Okay. Andrew was asking if what the link to the website that Juan was showing was, and just to confirm that that is not yet public, it's still under development, but we'll hopefully be able to share eventually. And question from Andrew, how do you deal with non-random sampling across the species range? Doesn't the kernel clustering tend to identify where there are points clustered?
Yeah. Yeah, we are when we have the data, we go through different process of cleaning data in terms of, for instance, overlapping data. We are removing those records, and we also have like some rules like that we are discussing those rules with Chloé and Christine, if those are the final rules in order to remove some of the records. That's one thing. And the other thing, we are also using a technique called spatial filtering, spatial thinning, that we are trying to remove points that are very close together. So in order to remove that kind of spatial autocorrelation among those points. So then we run the analysis to identify those clusters within the EBAR, and that's the density map that we have there. There are some discussions about why we need to remove points that are very close together, why not include those points, because maybe those points can just capture more information, especially for those species that are very rare and have points very close, but with different capturing different like environmental variables. So we are testing that also to see we just leave all the points without that spatial thinning or not. So we are testing that. This is something that came new just recently.
Great, thank you, Juan. Here's a question from Cassandra. Cassandra writes, I see that this initiative works both with observation data aggregators as well as some groups directly, such as CDCs. I know natural history museums are included in this process indirectly through aggregators like GBIF, but would the KBA initiative benefit from a more direct collaboration with or contribution from museums? And if so, what kind of contributions would be most useful?
I can get started on that question. There is currently museum data in our database, as you mentioned, through aggregators like GBIF and also directly. So Christine also reached out to various museum partners to gather data from them. And so they are involved in that way, but certainly we would benefit from a more direct and more in-depth collaboration. There's always, as we were saying earlier, room for more partnerships, and we're certainly interested in welcoming anyone who would like to be involved. In terms of museum contributions, the items that come to mind as that would be particularly useful are, of course, more data that may not be in aggregators already, and then also certainly expertise about certain species and sites that can be brought to us either through museums' involvement in the actual KBA identification process or their involvement in the review of KBA sites during the technical review stage. So certainly there would be room for that, and that would be highly welcome.
Great. Next question comes from Sarah. Sarah writes, are KBAs in Canada considered other effective area-based conservation measures, OECMs, by the CBD, i.e. have they contributed to the Canada reaching a high target 11 or target 1 for Canada? KBAs seem miles ahead of protected areas in terms of strategic biodiversity conservation, and they seem like great OECM candidates. And thanks to you all for your presentations.
Thank you, Sarah, for the question. I can also attempt to answer that. Because KBAs don't come with specific management prescriptions, they're not considered other effective area-based conservation measures. So though certainly they would be good candidates for OECMs. And the identification of KBAs is part of the Pathway to Target 1 process, in the sense that there has been interest from the Canadian government to use the results of KBA identification processes to help prioritize protected area designation in the future. So I hope that answers the question.
Okay, we have a question for Christine from James. What is the general size or scale of the eco-shapes used?
Yeah, so we're looking at the eco-district level, which is about a 1 to 50,000 scale. And if you'd like more information about exactly what layers went into the eco-shape mosaic, they're actually all found at the end of our methods document, and you can find a link to our methods document on our website.
Thank you. Next question from Rachel. If a site is already being considered as a KBA, is there a timeline for when a decision will be made?
So if I understand correctly, that would be kind of the timeline from the moment where we start the identification process to the actual formal recognition of the site as the KBA. That timeline is going to be widely variable depending on the site. There are many steps involved in the process, from the actual proposal to the review process, etc. So I would be hard pressed to give a specific timeline, but I would say it ranges anything from like perhaps a month to several months. And then of course it has to be reviewed by the Canada KBA Secretariat and also by the Global KBA Secretariat. I guess we should say also that these KBA sites will be reviewed every 8 to 10, 12 years. So there's also a system in place to have the information reviewed on a regular basis.
Mm-hmm. I'll just add to that there, as Chloe said, is a pretty stringent review process for each KBA that it must undergo. And we're still working on testing out and developing some of the processes that we're using for that review process. So there are sites that are really close to reaching the end. There's one site that has been confirmed as a KBA, which is Trial Islands in BC. But hopefully we'll have a whole batch of sites available soon. So this is also related to the Pathway to Target 1. Chloe, I don't know if you want to say any more. I think you've already answered it. That the federal government's committed to protecting 30% of Canada by 2030. They should be using KBA data and selecting those sites. And are we in communication with those people?
We are. Jamie, you might actually know more about this than I do, but there is involvement from Climate Change Canada in particular, and they have expressed much interest in using this information to prioritize those 30%.
Yeah, that's right. We're in contact with them throughout the review process, and they've shown great support for the initiative. Okay, Christine, how is EBAR used in BC and mountain terrain, i.e. using climate system maps? I don't think the intention is for these to be ecosystem level assessments. Is that right? Still a species approach, but bounded or context of evaluation is through the lens of jurisdiction specific ecoshapes.
Yeah, so I guess, EBAR is used in British Columbia, and, obviously we have our EcoShape mosaic developed all the way from Alaska down to Mexico. You are correct. We are not doing an ecosystem level assessments in terms of the fact that we're not creating EBAR range maps for ecosystems. These are species specific range maps. And I guess just in terms of the lens of jurisdiction-specific ecoshapes, I'll explain a little bit more that for Canada, because there is not an eco-district level range map available, what we actually did was we went to each individual jurisdiction and we asked them, what layer would you want to incorporate into our range map? And so we had that jurisdictional level consultation, whatever the jurisdiction asked us to incorporate is what we used. And again, to have more information about exactly what layer was used for each jurisdiction, it's all listed at the end of the methods document so that you can have that transparency and additional information. And Natalie, if you have any additional questions or I didn't fully answer your question, please feel free to send me an email and I can provide some more information.
Okay, let's squeeze in one last quick question from Peter, who says that these were terrific and informative presentations and wonders, are sources of community science information, so for example, iNaturalist, also incorporated into these tools, especially for the KBA scoping and EBAR? And if so, are they weighted differently at all?
Chloe, did you want me to answer that, or are you just muted? Okay.
Hello, sorry.
Oh, there we go.
Okay, start again. iNaturalist data is incorporated in the database. We have a lot of iNaturalist data, specifically the one labeled research grades in the database. It is being used by Christine's team in the EBAR mapping process, and it's also being used by our team indirectly through the EBAR maps and then directly as well during the KBA identification process. So typically that's data we use to help us delineate sites, to help us identify which species are where, etc. So we definitely rely on this data set a whole lot. And definitely add to this, Christine, if you'd like.
And do we weight that community science data any differently than other data types?
So mostly we weight element occurrence data as the most high quality, highest kind of vetted data. And so that's the data we use when we want a really high quality confirmation that species is present. And then other sources of data, including iNaturalist, are considered kind of less reliable than element occurrences, but they're packaged together in this second category of data sets that we use with a bit more caution. And the way we also ensure that we have access to the best quality data is through this filtering of research grade data. So we rely on iNaturalist data that's been labeled research grade specifically.
Great. Thank you so much, Chloe, for that response, and also to all of our presenters today. That's all the time we have. As I said at the beginning, this presentation has been recorded and we'll make it available on our website tomorrow. I'll also share it on Twitter and things, so you can also email me if you need a direct link. Thank you very much again, everybody, for attending, and we will see you in a month for our next webinar.